Hi everyone, we're Group 13. In today's presentation, we will introduce you to the Microbiota-Gut-Brain Axis and explore its impact on disease and immunity. Starting with a quick refresh, we learned from the lecture that the Microbiota-Gut-Brain Axis is a bidirectional communication system between the gut, the microbiota contained in the gut, and the brain. This intricate network involves a constant exchange of signals and information and plays a crucial role in maintaining the homeostasis of various physiological and psychological processes. Communication between the gut microbiota and the brain occurs through many pathways, including the immune system, autonomic nervous system, enteric nervous system, and the hypothalamic pituitary adrenal axis. The composition of the gut microbiota can impact signaling pathways along this axis. Gut microbes have the capacity to influence brain signaling through the release of various cytokines, metabolites, and neurotransmitters. And conversely, as it is a bidirectional system, signals sent by the brain can also alter the gut regulatory functions and microbial behavior in the body. Altogether, changes in any component of the microbiota-gut-brain axis can affect one's health and well-being by influencing their digestion, metabolism, immune functions, and even cognitive functions and behavior. Such changes can even contribute to the development of mental health disorders, neurodevelopmental issues, as well as other diseases.
Firstly, the autonomic nervous system plays a major role in the communication between the microbiota, gut, and brain. Signals travel between the peripheral and central nervous system, where the brain and the spinal cord takes in sensory information and respond accordingly. The sympathetic branch, more famously known as the fight or flight response, and the parasympathetic branch, or the rest and digest response, are both important in maintaining homeostasis of the body. Moreover, signaling from the gut can have major impacts on the rest of the body, with many of these signals coming from the microbiota. The microbiota actually produces many molecules that act as neurotransmitters, which can affect gut motility or even behavior and mood. The most important pathway of the nervous system for communicating with the gut is the vagus nerve, also known as cranial nerve 10. It is sensitive to changes such as the stretching of the gut and chemical changes, which then cause the brain to respond with electrical signals and hormones, such as neuropeptides for feeding. It is also important in regulating inflammation through the vagal-vagal anti-inflammatory reflex loop, which then reduces the levels of pro-inflammatory cytokines. Signaling through the vagus nerve is vital, and its disruption is implicated in many psychiatric disorders, including mood disorders. Of course, the molecules the microbiota synthesizes, like short-chain fatty acids, are important in this as well, with dysbiosis being associated with neurological diseases. The enteric nervous system is similarly a part of the ANS. It actually develops alongside the microbiota, each influencing the other. Pattern recognition receptors such as toll-like receptors activate and modulate the growing ENS, and the ENS has some role in controlling the microbiota population. Hence, the microbiota is important in development, and lack of it severely hinders the ENS. Additionally, neurotransmitters such as serotonin are made by the microbiota and affect motility of the gut via the ENS.
Another key non-neuronal route of communication is the hypothalamic pituitary adrenal, or HPA axis for short. It's also referred to as the stress axis because it's the main coordinator of the body's response to stress. For a quick overview, the process begins in the hypothalamic region of the brain. When the body perceives stress, the hypothalamus releases a hormone called corticotropin-releasing hormone. CRH travels to the pituitary gland, triggering the release of adrenocorticotropic hormone. ACTH stimulates the adrenal glands above the kidneys, which produce and release glucocorticoids, primarily cortisol, into the bloodstream. Cortisol is known as the stress hormone because its levels rise in response to stress and regulates various physiological processes, including immune function. A vital feature of the HPA axis is its negative feedback loop, in which cortisol inhibits the hypothalamus and the pituitary gland from secreting more hormones in order to prevent its overactivation. This regulation of the axis has been implicated in various health conditions, including anxiety, depression, and chronic stress-related diseases. But how is the HPA axis connected to the gut microbiome? Research on germ-free mice shows that the gut microbiota can influence the HPA axis, but also vice versa. Germ-free mice showed an exaggerated HPA stress response, which was reversed by administering the probiotic Bifidobacterium infantis. Altered expression of receptors in the brain, like serotonin, were also observed in germ-free mice. Additionally, in humans with irritable bowel syndrome, or IBS, microbiota alterations correlate with increased stress hormone responses. Moreover, the HPA axis interacts with the vagus nerve and the immune system. This image illustrates how chronic stress, more so than acute stress, can have detrimental effects and cause an imbalance in the microbial community. Elevated cortisol levels lead to increased gut permeability. In the leaky gut, native bacteria cross the epithelial barrier, resulting in the secretion of pro-inflammatory cytokines like IL-6. IL-6 activates the HPA axis and also downregulates glucocorticoid receptors, rendering the HPA axis hyperactive. Evidence suggests that an imbalance in the gut microbiome and the HPA axis is linked to psychological disorders like depression, autism, and schizophrenia, but more research is needed to further understand this complex relationship and discover alternative treatments to alleviate such symptoms.
The gastrointestinal tract is rich with immune cells, which communicate with our gut microbes through a layer of mucus produced by goblet cells. The microbiota provides metabolites to the host, many of which modulate the host immune system. The innate immune system comprises the body's primary line of defense against pathogens. The nervous system has its own innate immune system, composed of microglia, sensor cells that detect changes in the molecular environment and carry out a wide variety of functions. These functions include activation of neuroinflammatory responses, release of cytokines, synaptic remodeling, and recruitment of other immune cells, such as monocytes, which clear cellular debris from the brain. It was recently discovered that a diverse microbiota is key to the maintenance and maturation of healthy microglia. In germ-free animals, microglial populations are elevated in numbers and show impaired maturation and activation, as well as a compromised immune response to bacterial and viral infections. Recolonization of gut microbiota was shown to rescue this microglial phenotype. Probiotic treatment was also demonstrated to improve neuroinflammation-associated behavior through modulation of monocyte recruitment by microglia.
In addition to the innate immune system, the adaptive immune system is critically shaped by microbe exposure, and increasing evidence links it to homeostasis modulation of the brain. Studies in Drosophila have found social regulation through microbiota, maintenance, and immunoregulation. Some intraepithelial lymphocytes have been shown to require the presence of certain microbes in order to mature, demonstrating the interaction between adaptive immunity and microbes. In mice, impairment of the adaptive immune system is linked to alterations in cognition and anxiety-like behaviors. These abnormalities were corrected with microbe exposure. Although further research is needed, the link between the immune system and microbiome and the brain clearly impact many aspects of human health.
With all the different factors mentioned, the gut-brain microbiota axis can play a role in the development of mental health disorders. One such example is major depressive disorder. Major depressive disorder is correlated with an increase in pro-inflammatory cytokines. This change causes hyperactivation of the HPA axis. In addition, prolonged HPA axis activation impacts the trafficking of monocytes from the spleen. Hence, imbalance in one system can affect other systems. Depression can also affect other pathways that are related to the synthesis of dopamine, including glutamate and GABA pathways. Certain types of gut bacteria can break down GABA, resulting in changes in brain function and behavior. Poor diet is recognized as a risk factor for depression. Therefore, dietary improvements would involve increased consumption of prebiotic foods. In addition, supplements like polyphenols have shown potential in reducing depressive-like behaviors and mediating increases in pro-inflammatory cytokines. However, further studies are necessary to determine their benefits. The gut microbiota is critical for the development and maintenance of brain function. By further exploring the intricacies and impacts of the Microbiota-Gut-Brain Axis, we can find more approaches for mediating health and mitigating disease.